Excited to present on our recent work on inferring relationships among individuals, and we're doing it in a unique way that I'll get into in a moment. So first, I want to start with a little bit of background for those that may not be familiar. So in genetics, we tend to use figures like this on the far right, where there are individuals represented as circles and squares. Squares represent males, circles represent females, and horizontal lines joining a male and female represent a couple, with a vertical line dropping from this representing and connecting them to their children, and so on. So this plot shows a three-generation pedigree, and throughout this talk, I'm going to have shaded individuals shown where we consider we have data for the individuals that are filled, and beneath some of the individuals, I have two vertical bars that represent the two different chromosomes that an individual carries. So here, the grandfather has chromosomes that are two different shades of blue, and grandmother has red and orange chromosomes. And because each parent transmits only one chromosome copy to each child, the children each inherit half of their parents' chromosomes, their genomes, but it's a random half. And in fact, because of recombination, there's a mosaic process whereby this female child here in the second generation receives the dark blue chromosome from the father at the beginning, but then switches to the light blue, and so on. And in these figures that I'll show throughout, regions where two individuals have the same color are said to be identical by descent. That is, they will be the identical chromosome between these two individuals. So here, this child, this female, and her brother have identical chromosomes, indeed identical on both copies from the two parents, dark blue and red. So that's a bit of background, and then just going a little bit further. So in one generation, a parent transmits half of their genome. But if two relatives are separated by G generations, they're expected to share, on average, 1/2 of a proportion of their genome. So that's the expectation. Unfortunately, there's a fairly sizable variance in this quantity. But just to give you an idea, I'm not going to get into too much of this math in this talk, but just to give you an idea of the amounts of genomes shared between fairly close relatives, full siblings are expected to share 50% of their genome, but the 95% confidence interval is between 43 and 57% of their genomes. Aunt-nephew relatives share, on average, 25% of their genome, and first cousins share 12.5%. And as I mentioned just now, this fraction goes down by half in each generation that separates a pair of individuals. Okay, with that background now, I want to motivate relatedness as very important for genetics. So relatedness is needed in the context of genome-wide association studies, where we might have a set of cases, perhaps shown in black here, and controls shown unshaded. And when we do genome-wide association studies, we tend to compare the frequency of alleles between, let's say, cases and controls. But if there are more close relatives in the cases than there are in the controls, you can, in fact, get spurious associations to a phenotype, a disease, or some sort of trait that has to do with the relatedness among those cases as compared to truly biologically meaningful association signals. And so it's very important that we characterize relatedness as we conduct genome-wide association studies. Additionally, most population genetic models require that the analysis be performed on non-related samples. So we have to characterize relatedness in order to filter out the relatives from these. Also, in sort of popular genetics, there are several companies. This may not be exhaustive. I'm not intending to favor any of these companies, but there are several companies that offer genetic testing and will tell consumers about their relatives that exist in their database. Being able to better infer relationships is important for the general public, and is obviously of great interest to the general public as they continue to buy kits from these companies. And finally, there's an interesting feature of relationships is that as data sets grow in size, the number of samples with a close relative increases very rapidly. So, for example, in the UK Biobank, this is a data set that's recently been generated where there are roughly 500,000 samples from the UK. 30% of those samples have a first cousin or closer relative in the data set. So, the rate at which this occurs, the rate of close relationships grows very dramatically in sample size. So if we're thinking about, for example, population genetics, we need to be able to consider and model close relationships in these large studies in order to avoid problems of errors or of needing to filter extensively. We'd like to be able to merge existing models with models that deal with relationships appropriately. One more piece of motivation before diving into more details about relatedness and what we did. In addition to what I mentioned on the previous slide, close relatives are very useful for studies of recombination and of mutations. These close relatives have been used in many studies in the past, and they continue to be used to study these fundamental biological processes. And additionally, inferring haplotypes, otherwise known as phasing and imputation, can be done with near perfect accuracy in families because you can observe directly the transmission of alleles between parents and children or other very close relatives. Okay, with that now, I want to talk about relatedness and how to do relatedness inference well. So recently, we conducted an analysis where we compared 12 methods, state-of-the-art methods for inferring relatedness between pairs of individuals. And in this analysis, we found, so what I'm showing here are this is based on real data, where we have first-degree relatives, for example, parent-child and full sibling relationships are first-degree relationships, and second-degree, true, reported within this data set, this curated data set, true second-degree relatives, for example, half-siblings, grandparent, grandchild, and aunt-nephew relatives. And what we are looking at is the rate at which these methods infer these pairs of individuals as having first or second-degree relationships. So inference is in some sense along the y-axis here. And what you can see, these numbers are a little bit small, but what you can see is that most methods infer these relationships correctly at a rate of over 98% of the time. So first and second degree relationship inference is extremely accurate with a host of different available and commonly used methods for inferring relationships. However, when you go further out to fifth, sixth, and seventh degree relationships, so for example, fifth degree relatives include second cousins, sixth includes second cousins once removed, and seventh are third cousins. So if we look at these results, we can see that for seventh degree relationships, the most accurate methods have only about 40% of these true seventh degree relatives inferred as seventh degree. And so from this analysis, we concluded, among other things, that the IBD segment-based methods, which are shown with red colors for their names, perform best as compared to alternate strategies that don't detect these long segments that are shared identical between samples. And in addition, we concluded that perhaps quite obviously, it's best to try to infer relationships using the closest possible relatives from the data. And so from this, we had an idea, which is that we were interested in knowing whether we could try to reconstruct genetic information about individuals that we don't directly have data for. So what I'm showing on this slide is a set of 4 siblings and one of their distant relatives, where we are showing the blue regions are regions where these siblings are IBD, or identical by descent, to this distant relative. But of course, even though each of these siblings has a different region that is IBD to this distant relative, they must have inherited those segments from one of their parents. And as a result, we can just simply take the union of these IBD segments to infer the IBD sharing profile of their parent, in this case the mother, to this distant relative. And this is very nice because it starts to combine information from multiple individuals rather than only considering pairs of samples to infer relationships. It combines data in an effective way, and moreover, it removes one generation of distance between this relative and the individual that we're trying to infer relationships with. So in this case, we would try to infer relatedness between the mother and this person. And as I'll show you in a moment, this turns out to be very effective. And I'll mention that on the previous slide, the methods that that we had done analyses with were all pairwise. So this is moving in a direction that has only begun to be analyzed and has great potential. So the method that we've developed is called DRUID, which stands for Deep Relatedness Utilizing Identity by Descent. And this is work that was led by Monica Ramstetter, who was a PhD student in the group. Okay, so just a little bit of details on how this works and how we infer relationships. So if we consider 2 samples, S and D, where we have data for these 2 individuals, we can derive a coefficient of relatedness by calculating the length of the IBD regions that are shared between S and D and dividing by the genome length. This just gives the proportion of the genome that is shared IBD between these 2 people and can be used to infer a degree of relatedness. So this is how this sort of inference works when you have data for these 2 individuals. But if we had some set of siblings, S, we could infer the coefficient of relatedness between their ungenotyped parent, P tilde, and some distant relative, D, that we have genotype data for, by taking the union of the IBD regions shared between each sibling and D, and again dividing by the genome length. Now, an additional factor that exists in this equation as compared to the above is this proportion of the genome that the parent, P tilde, transmitted to these children, these siblings, S. But of course, this number is something we can just move to the right-hand side and end up with a coefficient of relatedness between P tilde and D. Now, what this leaves unspecified is what is this proportion of the genome that the parent transmitted to these siblings. Now, we looked into trying to estimate this transmission proportion by analyzing data from these siblings, but it turns out that we just ended up using the expectation of the amount of the genome that a parent should transmit to S, a set of siblings S. And this is just 1 minus the probability that a set of siblings of size, S bar, the size of S, the probability that those siblings share a haplotype with each other identical by descent. And so that's just 1 over 2 to the number of these siblings is the probability that they share one chromosome copy perfectly identical with one another. And then if we take 1 minus this, that's the proportion of the genome that the parent transmitted to these siblings. So this gives us our coefficient of relatedness for an ungenotyped parent of a set of siblings and allows us to do relatedness inference. So now I'm going to show you a set of results throughout this talk, throughout all of these results are based on simulations, but we have real data in the paper that recapitulates these findings very closely. So here what we have are a set of, in some sense, there's an x-axis here where we have between 2 and 5 siblings that we use in our analysis, and then there's implicitly one distant relative that we're computing the degree of relatedness to. And what we're comparing to is a method that I'm calling refined IBD. This is just based on the coefficient of relatedness from the previous slide, where we tally the IBD regions between each sibling and the distant relative D, and then we average the proportion of individuals that are inferred to have the given degree of relatedness to that person. So all of these numbers are the proportion of siblings in our set S that are inferred to have the correct degree of relatedness. So one thing that is immediately clear, first of all, DRUID always outperforms refined IBD, and as you add more siblings, so as S gets larger, the inference rates, the correct inference rates of DRUID increases as you go, in some sense, from left to right. In addition, so thinking about when S is very large, when we have 5 siblings, a DRUID tends to infer between roughly 16 and 20% more samples correctly than this pairwise state-of-the-art relatedness approach, refined IBD. And this is absolute terms. This is not 20, an average of 20% more, but in absolute terms, there are 20% more samples that are inferred correctly. So this is very effective, but we'd like to be able to do even more. So far, I've only been talking about using information from a set of full siblings. And the reason full siblings are used is that it's actually quite easy to identify full siblings for the reasons I'll talk about right now. So for considering first degree relationships, we have parent-child and we have full sibling relationships. Parent-child relationships are distinguished in that a child shares one chromosome copy, IBD, on exactly one copy with their parent at each position along their genome. But a distinguishing feature of full siblings is that there are regions where both of these individuals share both chromosome copies identical with one another. So shown here, highlighted in this box, are at the top of the chromosome. Again, these two siblings share a dark blue and a red region identical, and then in the middle there's a light blue and an orange region that are both inherited by these two siblings. And this is a distinguishing characteristic that should, in non-inbred individuals, should only occur in full siblings, these IBD2 regions, they're called, IBD on 2 copies as opposed to 1. Now, again, so it's very easy to distinguish full siblings using these IBD2 regions, but we'd like to be able to use more individuals. And so we could consider trying to expand to second-degree relationships, half-siblings and so on. But the trouble is, it's very hard to distinguish these second-degree relationships from one another. And we need to know the relationship between all of the individuals that we're taking the union of their IBD with in order to know which ancestor we're inferring an IBD profile are, which person that is. So we'd like to be able to distinguish these individuals, but unfortunately, current methods have very high error rates. However, we had the insight that it is possible to distinguish aunts and uncles of a set of 2 or more siblings by leveraging the fact that an aunt or uncle of a set of 2 siblings necessarily shares IBD2 regions with the parent of those 2 siblings. So we can detect regions of IBD2 between the ungenotyped parent of 2 siblings in the following way. So we first look for regions where these 2 individuals, these 2 siblings, are not IBD with each other. And these are regions where they necessarily inherited the 2 different chromosome copies of their parent. And simultaneously, we find places where those non-IBD regions are also both IBD with this potential aunt or uncle. And these are necessarily locations where the ungenotyped parent of these siblings is IBD2 with the aunt or uncle, because there's two distinct chromosome copies in these two children derived from that parent, and those two distinct chromosome copies are IBD with this second degree relative, the aunt or uncle of these full siblings. So we use this idea to try to identify aunts or uncles. And in this case, I'm showing real data for aunts, uncles, grandparents, and half siblings. And we quantify these regions where the two siblings are IBD 0 with each other, and then have an IBD segment with the aunt or uncle as IBD 011. So our analysis looks for total amounts of IBD 011 of greater than or equal to 50 centimorgans. And if you don't know what that unit is, there's roughly 3,000 centimorgans in the human genome. So it's a fairly small fraction of lower bound to infer aunts or uncles. But as you can see here on the right, only aunts or uncles as compared to grandparent, grandchild, and half-sibling relatives really have substantial fractions of this IBD 011 quantity. And in fact, DRUID is able to identify more than 92% of real aunts and uncles using this approach and gets 0 false positives. By doing this, by identifying aunts and uncles with such high accuracy, we're now able to infer the IBD profile of an ungenotyped grandparent of a set of siblings, right? So we take the data from the siblings, take the union of that, and union that further with any aunts or uncles that we detect by these means, and that gives us an IBD profile of an ungenotyped grandparent. So now I'm going to show results from this approach. So just to orient you, once again, we have different numbers of siblings, as shown here at the top, 2 through 5 siblings. And refined IBD is once again just a pairwise analysis, but DRUID is allowed to use 0, 1, or 2, color-coded as red, orange, and yellow, numbers of aunts or uncles in this analysis. So one thing that's immediately clear is that as more aunts or uncles are included, DRUID does better. And this is as expected. I should mention that the accuracy results here are only for the siblings, only for the bottom generation, to make it more directly comparable to refined IBD. We only use that last generation. And again, so as the aunts or uncles are included, DRUID does better. And with very large numbers of siblings and 2 aunts or uncles, DRUID can infer almost 70% of 7th degree relatives, so these are third cousins, correctly, as compared to only 36% with this pairwise method, refined IBD. So overall, DRUID can infer roughly 30% or more pairs of relatives correctly by using these aunts and uncles as compared to a pairwise approach. Okay, so in addition, and I'm not going to belabor this point, so in addition, DRUID can in fact not just use one distant relative in its inference, but if you have 2 different clusters of close relatives, it can infer and do relatedness inference using the IBD profile that's reconstructed for 2 ungenotyped individuals. So both sides can in some sense be ungenotyped in order to do an analysis. And with this sort of approach, DRUID now becomes something that we can compare to the state-of-the-art method for multi-way relatedness inference, which is called PADRE. So PADRE is a method that uses a composite likelihood approach based on the output of URSA and Primus to compare the likelihoods among different possible degrees of relatedness between 2 different clusters of close relatives. So again, this is more directly comparable to DRUID and is, again, a multi-way relatedness approach. So I've, in some sense, flipped the axes compared to the previous slide. So what we have here is just results for either 2 siblings or 5 siblings. And we have, once again, inclusion of either 0 aunts or uncles, so this is just 2 different sets of siblings that are distantly related to each other, or 2 siblings where each side has 1 aunt or uncle, and again, siblings, 2 sets of siblings where each side has 2 aunts or uncles. And one thing that, once again, as expected, these methods do well as more aunts or uncles are included, but overall, DRUID does quite a bit better than either refined IBD or PADRE, and is very effective at inferring degrees of relatedness, even out as far as 10th degree. So again, this requires a fair amount of data, but if you have data for 2 siblings and 1 or 2 aunts or uncles, DRUID can infer more than 40%, 35 to 40% of these aunts or uncles, these distant relationships, correctly. And once again, 10th degree relatives are 3rd cousins once removed. So we're pretty excited about this. And with that, I will conclude by saying that I've told you about a new method called DRUID that reconstructs the IBD sharing profile of an ungenotyped ancestor of a set of close relatives. This method can also infer and detect aunts or uncles of a set of 2 or more siblings with 0 in our analyses, 0 false positives. Compared to refined IBD, DRUID infers up to 18 or 30% more pairs correctly, and compared to PADRE, it can infer up to 30% or even more pairs correctly. And we're really excited about the potential for applying this to large cohorts where there will be data for siblings and aunts and uncles and those sort of things. So with that, I want to thank Monica Ramstetter, who led this work, and also Sushila Shenoy, who conducted some analyses that I wasn't able to get to, so I encourage you to look at the paper, and also my collaborators, Jason Measy and John Blangero. Thanks.